of the following exhibit a slightly different capacity in the effector function. However, the following is the important function, and the immunoglobulin G, the main function is it activates the complement like immunoglobulin M, that is also activating the complement. The more potent is immunoglobulin M, then acts as the opsonin and enhance the phagocytosis. So this will act as a opsonin and enhance their phagocytosis. They can also neutralize the pathogen and toxin, and they can mediate the antibody dependent cellular cytotoxicity. So what is the functions that is present for the class switching to the immunoglobulin G is, number 1 is activate complement, number 2 is acts as opsonin that enhance the phagocytosis, number 3 is neutralize the pathogens and the toxin, and number 4 is the mediated antibody dependent cellular cytotoxic and kill that cell. So the function of immunoglobulin G is very, very important because it activates the complement, it acts as the opsonin, it neutralizes the pathogen and toxin, and mediates the ADCC and clears the pathogen. One important function is IgG is also actively transported across the placenta by the receptor mediated transport, and thus play a crucial role in the production of the fetus during gestation. So they transfer through the placenta. Immunoglobulin G transferred through the placenta, and immunoglobulin A come through the breast milk. So these are the preformed antibody. Say this is the natural but passive immunity that is transferred to the baby. So immunoglobulin G, that is transported across the placenta by receptor mediated transport, and thus play a critical role of protection of the fetus during gestation. Okay, so this is the antigen binding site. That has we have discussed about the variable region, constant region, then variable region, constant region, heme region, complement. We have discussed about everything. Now let's move to the immunoglobulin A, that is the class switching to the immunoglobulin A, and this is occurs when there is the, I have told about this, the class switching of the immunoglobulin A will occur when there will be the immunoglobulin A, and when will occur? This will occur only when there will be the interleukin 5 is there. So interleukin 5, that will lead to the class switching to the immunoglobulin A. So another isotype of the antibody that can produce following the class switching is immunoglobulin A. This is more commonly produced in the submucosa than in the lymph node and the spleen. IgA generally exists in the dimer held together by J chain, that is produced immunoglobulin M. IgA has the following characteristic. So in the case of the IgA, there is the two things that you have to remember. In the normal, they are in the mucosa, they are present as the dimer, but normally they are as the monomer, similar to as the that of the immunoglobulin M, that was monomer initially, but in the secretory form they will present as the pentamer. In case of immunoglobulin A, that will be monomer, but in the secretory, in the mucosa, it will form the dimer. They serve as a major protective defense against the mucosal surface of the body. Any pathogen that infect the mucosa will invade the immunoglobulin A production by the secretion of this TGF-beta and interleukin 5. The major function is neutralizing the antibody by inhibiting the binding the toxin or pathogen to the mucosa of the digestive, respiratory and urogenital system. So where there is a mucosa, in the mucosa there form the layer paste of this antibody will be present over there. The paste of this antibody lines the mucosal surfaces, and then there provides the protection against the infection and the bacteria, and any bacteria that has IgA protease that can break down this immunoglobulin A will only cause this respiratory tract or urogenital tract or say GI tract infection. So any bacteria that is causing a respiratory tract infection, then you have to understand they have to bypass the layer, the paste, the layer of immunoglobulin A that is present on the mucosal surface, and that will only be possible when they have the IgA protease, that is enzyme that can produce this immunoglobulin A. So then only the infection of the respiratory tract will occur. So does not activate the complement or opsonin or mediate ADCC. So it cannot activate complement or act as opsonin or can do ADCC. These two types is there, immunoglobulin A and immunoglobulin A1 and A2. So they are normally present in a dimer in case of the secretory form, and secretory A, which is released across the mucosa of the respiratory tract, digestive tract, and urogenital tract, differ from the serum immunoglobulin A because serum is monomer unit in important fraction, as the A is dimer is produced by the plasma cell and B lymphocyte. It becomes bound to the poly Ig receptor on the basolateral side of epithelium endocyte, and then is based on the luminal bound to the secretory pieces that is received to the receptor. So the secretory component is dimer that is transported, endosized and transported to the mucosal membrane, that you have to understand. The secretory component thus serve as important function in the transepithelial transport, and once the lumen of the tract has function in protecting the molecular from proteolytic cleavage. So important thing is that you have to understand, this is the picture, plasma cell, they release the immunoglobulin A. The immunoglobulin A is now become dimer. They have been poly immunoglobulin receptor that has been dimer is internalized and cross the placenta, cross the cytoplasm, and then releases as a secretory immunoglobulin A in the mucosal surfaces, and they are actually secreted in the breast milk. Okay. Now, and provides the immunity to the baby. Now coming to the class switching to immunoglobulin E, we know class switching for immunoglobulin E, you require that is what is required, and that is required is immunoglobulin E will be due to interleukin 4. We can remember ever, E for EVER. So ever. So if E immunoglobulin E, that is interleukin 4 word. So in this way we can remember. Okay. So, but you have to understood this is the fifth letter, and fifth letter is not causing the proliferation of this immunoglobulin E. That causes proliferation of immunoglobulin A, class switching of immunoglobulin A. So IgE bind directly to the FEC, the epsilon receptor present in the mast cell, eosinophil and basophil, and involved in the initiation of the protective immune response against the parasite and allergen. So they have the role on against the parasite and allergens. It does not activate complement or act as the opsonine. So this doesn't activate the complement, this does not act as the opsonine. It heavy chain is called the epsilon chain. So this is the one of the classical pathway of complement activation which we have talked about. This is the bacterial surface. Here is this is antigen. Antibody has come that has activated the complement pathway. That is then C4 and C2 is breakdown. They forms C4b2a convertase. This is C3 convertase that will again break down this C3 into 3b and 3a. 3b will come. Then 4b 2a 3b becomes C5 convertase that will again break down C5 and form the C5b 6 7 8 9 complex, that is the membrane attack complex that puts the holes in the membrane, and C3b will be deposited excess on the cell and particle on the surfaces and act as the opsonization. So this is the classical pathway. Talking about the biological function, habits, and you know we have IgM, G, A, D, and E. These are the 5 types. Among that, mu for IgM, G for gamma, A for alpha, D for delta, and E for epsilon. They are the level of antibodies to less than 250 IgM, less than 350 IgA, less than, that is double, 600 to 1400 IgG, and there are trace amount of D and E. Function, the complement activation will be done by the IgM and IgG. Neutralization can be done by the IgM and IgG and IgA, but not others. Opsonization only IgG. Antibody driven cellular cytotoxic only IgG. There are some role of IgE also. Placental transplant, this placental transport is only possible from the IgG. Naive B cell antigen receptor, that is M and D. Memory B cell is present in case of the G, A, and E. Triggers the mast cell granulation by immunoglobulin E. So these are the functions that you have to remember of immunoglobulin. So we are coming to end of the chapter. There is a one term, clinical correlation, immunodeficiency involving the B lymphocyte. Patient with a B cell deficiency usually present with a recurrent pyogenic infection with extracellular pathogen. So we know if there is a patient with a B cell deficiency, B cell role is presenting antibody, that is the humoral antibody, and that humoral antibody is protecting for extracellular pathogen, and the intracellular pathogen is usually killed by the cell mediated immunity, and that is related to the TS1 arm, and that is a function of a T cell. So T cell activity, the intracellular pathogen will be killed easily, but extracellular pathogen will not able to kill if there is the B cell deficiency. So T cell system will be intact. T cell activity against the intracellular pathogen, delay hypersensitivity and tumor rejection are normal, but the extracellular pathogen that is dealt by the TS2 arm, or say the B cell, if they by secreting antibody, if B cell is deficient or say abnormal, then antibodies will not be secreted and extracellular pathogen will be not be killed and will suffer the disease from this extracellular pathogen. Hope you have understand about humoral immunity. Thank you.